Hello and welcome to the Deep Sea podcast, Pressurised, a short, punchy version of our main feed that gets right to the scientific point. If you like what you hear and you'd like to hear the full episode, you can find it in the same feed. And now, to get right to the point.
The guest today is some sort of scientific sorceress who does magic science with water. That's how I would explain it.
It's homeopathy.
Yeah, basically, yeah.
Water has a memory, and she can take a tiny bit of memory out the water.
Yeah, that's it.
Okay, let's talk to Georgia.
We're talking eDNA. So joining us today from Old Mate's office next door is Dr. Georgia Nestor, who's going to talk about eDNA. So let's kick off with what exactly do we mean by eDNA and how do you do it?
So environmental DNA just refers to any genetic material that's left behind in the environment by organisms. So we can capture that genetic material, or that DNA, and then see what species were in the environment at the time that we took a sample. So because it degrades over time, it sort of provides like a here and now view of what was in the ecosystem at the time that we took the sample.
How long is the DNA detectable in the environment after the animal is left?
So it depends really heavily on the environment that you sample. So if you were in a really nice cold environment, the DNA is preserved a little bit longer. If you're in like a really tropical, hot environment, it degrades a lot quicker. But modeling has been done. We've sort of seen that it's about anywhere between a few hours to a day max.
So is it easier then to do it deep sea than it is to do it coastal and shallow water because of the high pressure, low temperature thing?
Not really, because of the low biomass. So in the tropics, there's a lot of fish and there's a lot of species, there's a lot of corals, there's a lot of DNA. In the deep sea, however, it's a little bit more barren. So we're trying to just grab as much as we can out of that environment. So, you know, we might see like a few fish in our videos, maybe 10 max, and I'm just struggling to amplify that DNA up as much as I can.
And what causes the breakdown? Is it microbial? Is it a delicious little snack? Or is it that it's such a fragile molecule to be knocking around without a cell to protect it?
Yeah, it's a combination of all of those things, really. So we, with environmental DNA, detect generally people are trying to move on to larger fragments, but really we're targeting those small degraded fragments because of how quickly they break down. Essentially, it'll be released as a whole cell and then it just starts to degrade, where it be microbial or it could be UV or temperature. All those things start to degrade the cell. And then we'll target those tiny little fragments that are left over. We generally target mitochondrial DNA because it lasts a bit longer as well.
So for a bit of context, I should probably explain that Georgia works at EWA and is one of the regulars on the ship that which we operate around the world. And so the eDNA research is a major thread in what we do. So now would be a good time to explain the mechanics of how we get the sample and what the sample is.
For our deep sea eDNA program, we generally use our lander systems. On each of our lander systems, we have a Niskin bottle. The Niskin bottle on the surface is rigged up with a galvanized link. So when it gets to the bottom after a certain amount of time, that link will degrade and then the bottle will snap shut. So we're hoping that the water that it captures is like only deep sea water. So it shouldn't be any of the surface water or the mid-water column. I also attach some sponges and filter papers to the side of the landers as a passive method of detecting eDNA.
What do we call them again? The puffs? So.
The proofs, we call them. Yeah, so the sponges we call the proofs just because it sounds fun. But they are I can never say the word, but they're a poly-something sponge.
So this is one of these things that's interesting because there was a paper written not that long ago talking about how glass sponges are the perfect platform on which to study eDNA. Because it is a sponge, and that's exactly what we're doing. Do you have any data from those things yet to show that just cable tying a sponge to a lander is a solution? Because that's one of the greatest, cheapest sampling instrument attached to the most expensive thing ratio.
I guess when eDNA started, it was sort of like we can use it and we can do this water. And then I think over time, people are like, what if we just chuck a sponge on things? Or like, what if we, I was on a paper recently and it was like, what if we put a paint roller down a column? Will we see the same thing?
A paint roller?
Yeah, we were rolling pylons. It worked really well, actually, for benthic stuff. But with the sponges and the passives and the actives, because they work in such a different way, so the actives, we're actively pulling through 12 liters of water and we're capturing all that DNA on the top. I kind of was hoping that lapoufs or the sponges would not work because they're a pain to do.
That's just what.
Yeah, that's just where I was like, hopefully this doesn't work. This is awful, because we were pushing it through a syringe after each time. But interestingly, what I've seen so far from the Trans-Pacific stuff is each method captures a different portion of the community. So I was seeing a lot more of the diel migrators in the passive stuff because they're going down from the surface all the way to the bottom and then all the way back up.
Wouldn't the solution then be to do both?
Yes. So going forward, we're going to do it all. We're just going to keep doing everything. And if anything, I'll just attach more things just to see what happens, because curious. And
so this is like a bit of a loaded question, because I know the answer to this, but this is the thing I find most interesting about having watched you and your colleagues do eDNA for hours and hours on end over the last 2 years, is that it's probably the least immediately rewarding way of sampling the deep sea.
yes.
All you get is a bucket of water at the end and some little filter system that you got to freeze at the end. And we're all sitting next door with videos of sleeper sharks.
Yeah.
It's like, woo, look at our one species.
It's very interesting also to show people, like, you know, the crew will come down and be like, oh, wow, cool. So it always sounds so exciting. You get DNA from the water. What are you doing? And then they'll come and watch me filter water and they're like, see ya. That's awful. That's interesting, but no thanks. I'm going to go look at the fish. But I think not very instantly rewarding, but the data that you get out of it is incredible. So I should probably explain that with eDNA, there's two routes that you can sort of go down. One is the species-specific route. We can look at like the genetic variability within a certain species and just target one taxa. Or you could use what I generally do in our deep sea stuff, which is metabarcoding, where you'll apply certain types of primers to amplify up as much DNA from as many taxa as you can, just to see what's there. So then afterwards, you know, you guys will have however many fish you see on a video and whatever you see, and then I'll have 1 billion species to look through and see what makes sense and why they're there.
Yeah, that's what worries me. I remember you showing me the data from some of the previous trips, and it's just
Excel tables for days.
Yeah, it's just insane. But you know, getting the sample is actually the easiest bit in all this, I think. Then there's the boring bit of filtering it. Then there's like, oh my God, look how much data we have. But then there's a little problem, isn't there? Because we haven't got a reference library of every species in the sea. So then when these returns come in, they're only flagging to the nearest thing we have already in the global databases, right? So there's this issue of we might know how many species there are, but we don't necessarily know what they all are.
Mm-hmm.
So how do you deal with that?
The way that eDNA works, like you said, is we take our samples, we bring it back to the lab, we extract them, and then we'll amplify up the DNA through qPCR, and then we sequence those samples. Then it's a bit like forensics, where you'll compare it to a database of all known taxa at that time, or, you know, all sequence taxa, I should say. But like you said, there are huge gaps, especially for deep sea taxa, just because it's hard to get the tissue. When it comes back to the surface, it's generally, in a lot of cases, not the best quality to curate a whole genome, for example. So we might have only certain genes available for certain species. So what we do is I use a lowest common ancestor approach. So we might hit a Barythrides, for example, but we don't know what species it is. And I will pull that back to a genus level because only one of those Barythrides has a 16S gene available. So then I'll say, well, I can't confidently say which one it is because there's no reference material available. So I'll pull it back to a genus level. But the more that we sample and the more that we use tools like ROVs to actually pull up the taxa and barcode them, the better these reference libraries will get. And the good thing about eDNA is you can go back, kind of sounds like nightmare fuel, but you can go back and look at your data again and re-blast it years later and be like, oh, wow, look at how all of it's changed. Let me write another paper on that.
So does that mean you don't have to go to sea anymore? You just live off the current data set for the next 15 years?
I mean, you could, but we'd only ever find out about the Trans-Pacific. So...
Well, I think it's interesting that the one of the problems we have, deep sea, let's use deep sea fish, usually one of Thom's favourites, is the mighty Basozetus is nothing short of an absolute pain in the backside, because we know there are multiple species, we can see there are multiple species, but they're really cryptic because they all kind of look a little bit samey, and we only get to see the ones on the land, for example, that are interested in prey on amphipods that come to the bait. And we very rarely ever see them in the sub. And there's hardly any samples ever taken because they're just deeper than most people have ever trawled. So if we were to do eDNA alongside that, we assume we'll get lots of Basozetus eDNA, right? Because they're right next to the bottle that's sampling it. But then, is there a way of interpreting the data from your stuff to say how many species of Basiliscus are there, rather than just being there's a big Basiliscus soup in the middle?
I guess there's two approaches, and this is actually something that I am personally interested in using eDNA for in future research. One would be developing a species or genus-specific primer for Basiliscus and then trying to get that variation over and over again and using it across all of our samples. Because once we've extracted the DNA, we've got the DNA until we've run out, really. So we can keep analyzing it using these specific primers to see the genetic variability between those taxa groups, or species, I should say, and then see if we're detecting different species in different areas, and then hopefully do some sort of network analysis with that. That's one approach. Or something else that I'm super interested in doing is a taxonomy-free approach. So we get these things out of our sequencing called amplicon sequence variants, or ASVs. I want to look at, okay, we're in the Pacific, we keep seeing this one amplicon sequence variant over and over and over again. It's hitting Basiliscus quite low. Then we move to Antarctic, we're getting a different amplicon sequence variant, but it's the same Basiliscus hit. And then comparing those and saying, well, we've gotten these certain sequences over and over and over again. They likely represent these different species or variations of the same genus.
Yeah.
That's something that I'm super interested in. But it would take a lot of machine learning and AI and Bayesian models and time.
But this is one area of deep sea science which is absolutely moving into machine learning and AI now, isn't it? Because it's just your volume of data that comes in versus the volume of data you have to blast against. It's becoming a automation tool, right? And trying to figure out the best way of dealing with this, because going through these things manually, line by line, going, which is this really a mictophoid or not, is not the most effective use of time. Plus, it would probably mentally destroy anyone who's trying to do it, which I'm sure you'll agree with.
Yeah, I mean, it's only made me stronger, but yes.
Has it, though?
No, mentally, I feel weaker.
Yeah, I've seen you slowly degrade over the last 2 years. Another big question for eDNA, which is, especially when you go into very deep water and you've got a period of time where you know that, this fragment has left an animal. How do we know that a species detected in the sample is from that depth and not something that should fallen down from above?
I think with pressure, it would be, if it had fallen a couple 100 meters, it'd probably be fine. But over such a huge gradient, it's quite unlikely that these small fragments aren't degrading beyond the point of us detecting them. So in some of my previous work, and I'm seeing it in some of the Trans-Pacific stuff, the things that really confuse the data are those diel migrators, the lanternfish, and all those ones that go, oh, I'm going to go to the surface now, and now I'm going to go to 1,500 meters, and I'm going to keep doing that over and over again.
Yeah.
They really mess with my data quite a lot. But with the Basiliscus and the Barophrydes, I generally only get those in those bottom water Niskin samples, and then comparatively in the passive samples, I'll get such a small signal from those. In previous work that I've done in submarine canyons, I did get a lot of hits in the surface to, I think it was Basiliscus, and I was very confused by that until, we had a chat the other day, and it was larvae. Makes more sense.
Yeah.
Because we were finding the larvae.
We should tell Thom about this, because Thom interviewed the people on the Blackwater diving episode.
Oh, it was such a cool one, yeah.
Yeah, I was showing Georgia that paper the other day, showing there's all these deep sea demersal. I mean, we've known about this for decades, but it's nice to actually see them.
Yeah, and I guess because this was my PhD, so I was a little baby, and I had a CTD, and I would do surface, 100, 200, 500, 1,000, and then bottom water. And I was getting them in the bottom water, nothing, surface. And I was like, oh, I've mislabeled these, or something has gone wrong. But then, it makes so much sense after reading that paper.
Yeah.
And it's a thing we know, but I don't know, we sort of knew it without really embracing it. And now that we've got the Blackwater divers and people are like diving with Basiliscus. Yeah, and the rat tails as well. They're all planktonic at the juvenile stage.
It's crazy, yeah.
So there's a little story I want to tell that is purely to wind up Thom.
Oh, fun.
And it ties in. The next question I was going to ask was, what's the most unexpected thing that this method has turned up? But don't answer that just yet, because I want to wind up Thom on this. So Thom just spent loads of time in Antarctica trying to find the colossal squid. But then I remember when the first time you came into my office and said, if you get me water samples, I can do eDNA. I'm like, yeah, whatever. And you showed me the stuff you did off Australia. And one of the things that jumped out was, without even trying, you found the giant squid off Australia.
I guess that's one of the super rewarding things about eDNA is when you look at the sequences after and you're Googling them and you're like, hang on. And then you pop it in and you're like, yep, that's definitely the giant squid. And it's super rewarding. It's nice because there's a lot of these species that they're monotypic. So it's not anything else. I'm hitting it at 100%. I'm getting these massive amounts of sequences. It's definitely there. And we're expanding species ranges and all those sorts of things. But I guess the downside is, you don't physically see it. So it's not as exciting.
It's hard for the press release.
Yeah, exactly. It's like, and we saw the giant squid in the DNA.
That's fine. This is just about getting one over on Thom because Georgia was on the same trip I was on in Antarctica. And so technically, in those samples that are currently in the freezer, we could have a little piece of colossal squid.
Mm-hmm.
But Thom
We
doesn't
had
have
all
that.
the primers, we had all the Niskin bottles, and a month and a half before we went out there, the permitting agency decided that they'd changed their mind, actually.
Really?
Yeah.
Really? For water?
Oh, and they wanted an ethics agreement because there might be animals in the water.
Nah.
Oh,
They-
they're not the smartest animals if they get in those Niskin bottles.
They totally undercut us. So yeah, we took 2 full-size, paid a fortune in extra baggage to take 2 full-size Niskin bottles and all the primers in an ice pack, and it was a nightmare to get that across South America. And then they pulled it out from under us right there.
Wow.
That was annoying. Well, you wanted to wind me up, Alan, but you didn't know how much you were winding me up because we tried.
It makes it even better.
Yeah, I guess I find that very interesting because generally eDNA, that's one of its benefits is that, we're only going out, we're only getting water, we filter it, we put the water back into the ocean. It's just, we've taken the DNA away, but it's quite non-invasive in that sense. Like we're not extracting animals. I think the only time was when a Dichorhynchoides or a rat tail got into the Niskin that one time, but that was a surprise. Apart from that, it's quite non-invasive. So I'm surprised, that you needed ethics.
I can honestly say there's been twice in my career that I've caught a fish using a Niskin ball. Yeah.
They're brilliant samples, though. They don't get much damage.
We had a snailfish in one off Atacama, which was perfect. And then I think it was it Tonga that there was a rat tail? It must have just been snuffing around. Was it Novacan? They're just snuffing into Niskin, and then just as it fired, and we just basically grabbed it by the head. It's so unlucky, it's unreal.
But great for barcoding, great for making a whole genome.
Exactly.
Yeah.
His death was
Plenty
not
of
in vain.
DNA in that whole fish.
Yeah, think of the DNA.
The wording sort of conjures this idea of loose, free DNA, like the actual molecule just loose and free in the seawater. Is that what you're mostly amplifying, or is it mostly individual cells that have been sloughed off?
Yeah, so it's mostly the free and the extra because whole cells, I think that's sort of an avenue that a lot of people are trying to go down is getting those whole cells to look at actual individuals, but it just degrades and splits open so quickly. And then the pressure in the deep sea, even getting RNA is quite difficult. So we target those small open fragments, really, those small broken pieces.
Hanging around.
Yeah, pretty much. Like it's a big old soup of just split up cells, really. Whole cells would be great for looking at individuals, but I don't want to say impossible, but they've been working on it for about 7 years now. And in a fishery where there's lots of them, and it's still very difficult to get a whole cell.
And are you seeing differences in the high pressure adaptations? Does deep adapted DNA hang around for longer? And do you have more confidence that shallow water DNA would be broken down more quickly by the pressure as it fell?
I used to think that. I just sort of thought, oh, that makes sense logically. But then based on some of the work that we've done and the scales that we do it at, in coastal water, I've done things in the Perth River here and at really small scales. And within the space of like 50 meters, the communities are looking different. Like we're seeing a breakdown within spatial scales. But then in the deep sea, I thought, okay, well, it's just maybe going to be a bit of a soup and it's going to be quite hard for me to get these spatial patterns in the data. But I've seen the same thing in the submarine cannons I did off the coast of Australia or Western Australia here. We would travel, a kilometre or two, drop another CTD, and that site was different to the previous site, which I found quite interesting.
Yeah, because you're watching all that marine snow rain down, and that must be full of the stuff. But if it's getting maybe denatured by the trip.
I think that is what happens because the difference between the 4,000 meter samples that I would take and the mid-water column was quite large. The 200 to 1,000 samples all sort of cluster together, which I guess makes a lot more sense. But then between 1,000 and 4,000 or 5,000, we're seeing these huge splits in the community and we're getting different structures. And I think a lot of that has to do with the benthic things that I'm detecting, obviously giving them a different signal. But yeah, we don't really get that many mid-water species coming down in the DNA. I did, like I said before, get some deep water species up on the surface, which we attributed to larvae.
What else have you found that's like completely unexpected? Because I guess a tool like this is supposed to illuminate all these rarities, like, you know, more so than we're ever going to see on the cameras.
Yeah, I think every time is a bit of a surprise. I know that's cliche and a bit boring of an answer, but I think I really like the gelatinous and the squiddies and all those sorts of lighty up pretty things. So I always find those in the samples. And I think because we don't see those as often on the cameras, I find that quite interesting and rewarding because, you know, unless they happen to go past our cameras, we probably won't see it. So the giant squid was very interesting. Also the Cuvier's beaked whale, and I've got a lot of quite rare whales that we don't see very often. And then especially with the Cuvier's beaked whale, I was getting that at quite a lot deeper than its max depth was recorded. So that's something that we could look into and be like, okay, well, we're actually diving deeper than we think. And is it correlating with some of the scratches that we're seeing on the bottom of the ocean? Is that from them playing around in the sand?
That's genuinely things we've seen. We've seen them in Nova Cairn at 3,000 metres, and we've seen the whale pits at, I think, also 3,000 metres in the North Australia Basin. So I think we had a conversation about this once that we can't, there's a bit of a so what factor to that. But at the same time, no one's ever seen Cuvier's beaked whale do it. They've only seen them come back on the surface over 3,000 metres of water with mud on their face. Apparently. That's
And I
Could
think
be some of that
that's,
pelagic mud.
yeah.
Yeah.
I think that's one of the good things about how we're doing it is pairing the eDNA with the conventional or the cameras and all those sorts of things, because it's hard for me as a genomics or a molecular ecologist to say, oh, well, it obviously is deeper than we think. Because people just say, well, that's DNA. You don't know for sure. It's probably sunk down. How would you know? I think we get a lot of flack sometimes for some of the conclusions that we might draw, which is fair because we don't actually see any of these processes happening. But then pairing it with the video and being like, well, no, because they saw these big pits on the floor. It makes sense.
Yeah, there's nothing else at depth that could make a pit that size because there are no big animals there, right? So it has to be a whale or something or whatever. But yeah,
Megalodon, yeah.
yeah, hopefully. We kind of mentioned a little bit machine learning and AI, but what are the next big things? What's the next big advancements in technology?
I guess on the genetics side, I really want to look at population structures with the eDNA and can we actually use some of these sequences, and hopefully if we get them back in, you know, the best preserved quality that we can, then we might be able to look at population health and structure using genetics and some conservation genomics, and then connectivity. But then I think advancing our methods towards maybe a taxonomy-free output is very beneficial. And then I think also, yeah, like we said before, the bioinformatics, machine learning and AI is going to be absolutely huge for this because we're just producing too much data. It's too much data for just a few people. We need to be able to train the data to plug it in and be like, okay, well, you're now in the Pacific. These sequences have never been seen here before. These ones don't make sense. These are always there, those sorts of things.
I really like the idea of the long-term stuff. So recently you've bought these in situ samplers that can take multiple samples over a long period of time. And we've got obviously the big long-term observatories off here. And what I'd really like to do at some point, whenever I get 5 minutes at least to think about this in more detail, but is to do something whereby we have a long-term lander down for periodically over years and take an eDNA sample maybe, I don't know, what, every 2 weeks or something like that. Because I still think there's a lot more migration that goes on, or at least we don't know if there's any migration going on or not. At depth. And I think this eDNA would be a far more appropriate way to address or at least investigate deep sea migration than cameras, because you see so little in the camera, especially long-term ones. When you take a photograph like every 12 hours or something like that, you know, the chances are there's going to be nothing in it. But it would be really fascinating to see how that community changes over time, because I still think there's a weird thing, and when I was discussing this with someone the other day about seasonality and how if these spring blooms and in the high latitudes all come down and there's this big pulse and all the rest of it, but there's also big, massive chunks, particularly in the Pacific, where they're sitting under oligotrophic gyres and nothing happens. But there's still stuff there. Now, why would a bunch of deep sea animals go to a place where there's no food all year round and there's no seasonality? Or the alternative to that is there's big migrations going on. And so the only way to answer that, I think, would be long-term eDNA monitoring. I think that'd be pretty cool.
A lot of people are starting up long-term monitoring programs. I think it's just, It depends on how much you can actually invest in these programs and what you want out of them. Because one of the benefits of eDNA is that you can get a whole ecosystem or like a tree of life view. You don't have to just target fish and animalia. You can look at bacteria. You can look at everything from the smallest of the small to the biggest of the big and target different groups. Once we get those samples back, you could use 25 primers if you wanted to amplify different groups of taxa and then try to look at that as a whole ecosystem view. And then, like you said, seeing those changes over time would be super beneficial.
It would also work for invasives as well, wouldn't it?
That's where it really started, eDNA 10 or 15 years ago now, was looking at invasive frogs and invasive things in river systems mainly, but it is heavily used, especially over on the east coast of Australia here. They have a lot of invasive monitoring using eDNA.
It monitors human health as well. Our sewage water is routinely tested, and they charted things like the spread of COVID.
Excellent. Anything else, Tom, you want to ask?
What do you use when you're running an analysis?
I do everything on a supercomputer. So, for example, the Trans-Pacific data that I got back, it was, I think, 400 gigabytes of data that if I tried to blast that on my own personal computer, it would, I think, immediately fry and die. So we have these Pawsey here in Western Australia, and then I'll upload that, write a script, or run it using one of our pipelines at UWA Oceanomics. And then when the data comes out on the other side, it'll be a nice Excel table that then I'll go through and then my computer will work.
Another room has gotten very hot somewhere.
Yeah, but it's not my room, so it's fine. It's a supercomputer one where they've got all the fancy cooling systems.
There's an ominous cube here. We sometimes get time on. Niwa has a cube.
They're always big black cubes. I don't know why. I think it is to make it look more ominous.
Well, with that, I'll say thank you very much, Georgia, for insights into eDNA.
Thank you for having me.
So how are you finding dabbling into the world of eDNA? When it comes to sort of interpreting the data, when it comes out the other end?
I really like it. Actually, there's some massive potential in it. And at the moment, it's still at the stage of trying to work out what everything is. You know, you're still kind of bit that library. And it's frustrating because our abundant species, our most encountered species, don't necessarily have have metadata already or genetic data to blast the new stuff again. So you do have to make a little bit of inference of, is that what we're seeing on the cameras, whatever. But the thing I do like is getting a real good, interesting snapshot of what we're not seeing. So there's a whole bunch of species we see on land. You drive around in the sun for 4 or 5 hours, you will not see a single one of them. So it's nice to sort of bridge that and expand that out into other ones. But what I really like is the questions you can ask about the cryptics. We are really struggling with things like Basilozetus, where you see it all the time and you know there's more than one because there's ones that just don't look right and there's ones that are white for some reason. And, you know, what the eDNA would do is at least give you some confidence. We don't have a sequence for any of these species, but we know from the eDNA, for example, there are 5 cuscules in that area. Or if we come back and say there's only one. There's only one cuscule gene in that whole area.
You know, you can be confident that now they're just really varied.
Yeah, so it's another really cool tool to throw into it. It's not one of these new methods that supersedes something else. I think it's another line of inquiry. At the moment, the biggest problem is the deeper you go, the less there is to. So when you get these returns and say, we think we've seen X, Y, and Z, you look at some of these like, no, you haven't. That's just the closest thing to it in the database. So there is a long, laborious kind of quality control on that. So you don't make ridiculous conclusions to it. The amount of data it produces is phenomenal. And so now we're at the point where, okay, we can produce huge quantities of data very, very quickly, but then you need someone like a bioinformatician to come in and start interrogating it.
It's an extra thread because ultimately we want to know what's there and what's going on. I don't like, obviously we get a lot of it at the museum. I don't like this like battle between different wings. Like everyone has the same goal. When they invented X-rays or when we got access to X-rays, the dissection crowd didn't get all like, oh, well, that's, you know, bullshit. You can't tell unless you pull the thing apart yourself. And then we got micro CT, and it's all just more tools. We're all after the same questions. So the more data we can get.
I think there was a big argument about 20 years ago with, are we going to lose our sense of natural history if we keep going down the molecular route? And it was like, it was never supposed to replace taxonomy. It was never supposed to replace filming an animal in situ, right? It's just that there's a lot of things we can do by filming them alive. There's lots of things we can do by examining them dead. And there's now lots of things we can do genetically. And eDNA is like just an addition to that. Now we can detect where they are without even seeing it or sampling it. See what I mean? So it's just this, it enhances everything else.
It's helping you interpret the visual.
Yeah.
And you still need the sniff test. Like no one went and executed all the experts. That you still need the sort of, yeah, but does this make sense?
If you think about things like the big fin squid and that whole thing we're talking through with Migrators, the depth and the geographical range of an animal like that is only very, very slowly being teased apart because you're waiting for someone to come across it by mistake. Process could have taken 10 years to make those little nudges as to where this animal is. Now, theoretically, you don't need to disturb it. You don't need to go looking for it. You don't have to wait for it to happen. Just sequence it in eDNA and then say, okay, the big fin squid lives all the way to Antarctica. Maybe. You know, it answers that question much more rapidly. But there are also questions about why the hell does it have such bizarre arms? That's not going to be answered by eDNA.
One big project we've been involved with is to have a really good database of at least New Zealand's fauna. And part of the advantage to doing it with that is there's a lot of stuff on the big databases which the specimen is gone. And if there's some doubt about its ID, or even worse, that species has changed since, then a lot of these databases, if you don't have a voucher specimen tied to them, you can't go back and look again, if that makes sense. So like if one species gets split into two and you go onto GenBank, well, which one do I have? And we threw it out. So we can't go back. And you'll be able to tease it apart, maybe genetically digging back into it, but you've lost that little bit of confidence. So you almost need the genetic holotype. You need the reference, you need the voucher to underpin those sequences so you can go back and check if things change. It's quite exciting stuff.
And that was a pressurized version of one of our longer podcast episodes. If you enjoyed that and you would like to hear the full-length episode, just match the episode numbers and you'll be able to find the full-length version in the feed. Thanks for listening. We'll deep see you next time, and I abyss you already.